Good morning. My name is Gabrielle Langlois, and today Grace, Lowry, and I are going to tell you about the research we've been working on this past semester on virus disinfection and regrowth that we did under Dr. Samuel Snow for the College of Civil and Environmental Engineering at LSU.
It is well known that ultraviolet light technologies are the most efficient at inactivating bacteria, viruses, and protozoa compared to chemical disinfection used in water treatment. When cells become exposed to UV light, cyclobutane pyrimidine dimers form in the DNA or RNA, which disrupts the pairing of bases during replication. This causes mutations that affect vital cellular functions. UV mercury lamps are commonly used in water disinfection, but the development of UV light-emitting diode technologies present many advantages. These UV LEDs require less energy, they're more compact, they have a longer lifespan, and provide UV light at any specific wavelength, which expands applications in water treatment. Using the various wavelengths that UV LEDs provide in water treatment systems introduces the concern of regrowth. A cell exposed to visible or UVA light ranging from 300 to 500 nanometers after disinfection can undergo photoreactivation. This is where photolysis enzymes use the energy from the light to repair damages done to the DNA or RNA. This process can reintroduce biological contaminants in the effluent after water treatment. Therefore, we aimed to study the effectiveness of disinfection technologies to inactivate viruses as well as the virus's ability to repair. We use 5X147 MS2 PR772 in our experiments as surrogates for common waterborne pathogens encountered in water treatment processes.
Similar to UV photoreactors, it's possible to use radio waves and electromagnetic field to heat nanomaterials in water and deactivate viruses. This is an alternate novel disinfection process which could potentially lack the regrowth issues we experience with UV because rather than UV, it uses heat as our inactivation mechanism. This method takes full advantage of our ability to manipulate virus particle interactions in solution and maximize disinfecting by giving us the greatest chance for disinfection by assuring that we have the most particles with their heat to interact with viruses. When iron oxide is placed in an electromagnetic field, it can generate high localized temperatures using low energy inputs from the radio frequency field. Viruses in contact with nanoparticles in a system like this are exposed to high localized temperatures on the surface of the particle and cause the DNA and proteins of the viruses to denature when they come in contact. Applying this disinfection method to our samples, we hypothesized that regrowth will be little to no issue with viruses in a system using heat as an inactivation mechanism.
In order to test the photoreactivation potential of the viruses, we performed a series of photoreactor experiments. To do so, a solution containing the virus in a sterile buffer underwent initial inactivation by UV light of 278 nanometers and 254 nanometers. The samples were then moved underneath a UV LED of 365 nanometers. Samples of the solution were taken before and after initial inactivation and every 2 hours during the time spent under the 365 nanometers. The samples incubated overnight on agar plates before we recorded the plot counts. To study the nanoparticles, we tested solutions of the virus in an electromagnetic field and controlled the radio frequency range to excite the nanoparticles and generate heat that we expect to denature the viruses. Post-experiment, we separate the viruses from the nanoparticles using the particles' magnetic properties so the particles could be used again. In effect, it will be similar to the photoreactor experiments, except we will be using radio waves instead of UV wavelengths. This figure represents the regrowth of E. coli from photoreactivation experiments previously done in this lab. We hope to find similar conclusive results for the viruses in these studies. We were unable to test 5X147 in these experiments, but based on previous studies, we expected to see regrowth. We had no initial expectations for regrowth of PR772 and expected no regrowth for the MS2 cells. Figure 1 demonstrates a log reduction of PR772 and MS2 versus time, which shows the effectiveness of inactivation. The initial inactivation under 278 nanometers or 254 nanometers took place from time 0 to 2 or 5 minutes. For the remainder of the time, samples were placed under 365 nanometers to see if the log reduction from the initial inactivation would decrease, showing regrowth. Virus PR772 demonstrated a slight regrowth of 27% between initial inactivation and 120 minutes. After 120 minutes, the regrowth of the PR772 cells either decreased or stayed fairly constant for the remainder of the experiment. MS2 had no regrowth except the sample that underwent UV light of 254 nanometers for initial inactivation. This sample demonstrated around a 26% regrowth between 240 minutes and 360 minutes. Figure 2 demonstrates the same initial conditions as before, except all 4 samples were placed in complete darkness after initial inactivation to observe the cell's ability to undergo dark repair rather than photoreactivation. Compared to Figure 1, we see less regrowth in PR772 with around 19% for both samples. The most significant regrowth observed was the MS2 sample initially inactivated by 254 nanometers with around a 34% regrowth from 5 minutes to 360 minutes.
For the first round of experiments, we used a commercially available iron nanoparticle with MS2. Initially, we were unable to observe any disinfection or really much of an impact, but we did notice some problems. During the attempt, we noticed that particles settle quickly to the bottom of solution. This is a problem because the chances of the viruses interacting with the localized heating on the particles is much greater when the particles are suspended in solution rather than settled and collected at the bottom. This is likely due to the large size of the nanoparticles, and even though the viruses did generate heat, it wasn't interacting directly with the viruses and causing disinfection. Some next steps we could take is we could strengthen the interactions by attempting to use a higher concentration of nanoparticles in the experimental solutions, or we could use a larger energy input from the electromagnetic field, which would generate higher temperatures, but this would be adding more energy, which is a little less sustainable. We can adjust the pH of a solution to possibly alter the surface charges on the particles and create a stronger interaction. The particles and the viruses both have a negative charge, so they weren't really interacting, but altering the pH, we could potentially change the surface charges on the viruses and particles and create a positive negative interaction, which would be really strong and give us pretty high chances of having the viruses experience this localized heating. This could maximize disinfection. Though we were unable to get as far into testing these theories as we wanted, we are providing important first steps in confirming the ability of the nanoparticles to disinfect and prevent regrowth. Further studies should take place on these particle interactions because they can lead to improving the photochemical activity and more efficient disinfection. The results of this experiment could help keep treated water safe from viral regrowth, which has harmful impacts on human health. The more we know about regrowth, it will help us better understand how to prevent the regeneration of potentially pathogenic viruses in our water distribution system. The use of electromagnetic fields as a power source for disinfection in combination with the iron oxide nanostructures can hopefully lead to a dramatically lower standard of energy input in the field of water treatment. Also, using these nanoparticles as an alternative to traditional wastewater disinfection technologies can help to avoid the production of hazardous disinfection byproducts, which are a common issue. We would like to take this time and thank you for your attention today, and we'd like to thank LSU Discover for giving us a chance to perform this research over the past semester and giving us the opportunity to share it. Thank you.